Hi, my name's Mattera, and I'm a grad student from Rutgers University in the Jim Simon lab, and I'll be talking to you about the history of breeding for basil downy mildew resistance in sweet basil. So the basil genus, Ocimum, is a large genus and has multiple origins of diversity from all around the world. Actually, the Italian sweet basil, um, the one that's culinarily famous around the world, is actually originates from Africa. So the importance of basil. Well, sweet basil is the most popular herb in the United States and in Europe and around the world. It's one of the most profitable specialty crops for small-scale growers. Thai, lemon, and many other basils are used and are very important in cultural cuisines around the world. And there's a huge diversity in basil, the genus, but also in terms of sweet basil, for phenotypes, the genetics, and chemotypes. So how they look, how they behave genetically, and how they smell. There's over 60 species in Ocimum, and just in Ocimum basilicum, in terms of leaf shape, color, aroma, and flavor, there's huge diversity. The basil genus, like I mentioned last slide, exhibits extensive variation in genetics, genome size, and ploidy levels. So this is actually advantageous because there's a high amounts of variation to select upon as a breeder, but disadvantageous because of the problems that come along with that in terms of sexual incompatibility, sterility, and unpredictable segregation patterns. So it can be difficult for a breeder like us to breed anything related to basil. So I'll point out two of these. Ocimum basilicum is a tetraploid and actually has 48 chromosomes, and Ocimum citriadorum, which is actually, I think now called Africanum, is a hexaploid and 48 chromosomes. So basilicum's tetraploid, citriadorum is hexaploid. They both have the same number of chromosomes, and that presents problems when we breed interspecifically. The downy mildew pathogen, which is basal downy mildew, which is caused by the pathogen Paranospora bulbarii, is one of the most destructive and fatal diseases of basil. It reached the US in 2009, and that's when it got on our radar, but actually was discovered in Uganda in 1930s. It overwinters in warmer climates, but essentially is now found all over the world, in US, Africa, Europe, and Asia. And there are almost zero chemical controls that are effective. There's some new chemistries out there that have actually shown some promise, but it's still very limited. And there are no viable options for organic production. And at the time our group set out to develop resistant basils, there were no commercial resistant sweet basils available. And this disease can lead to 100% crop loss. And here's a picture of what downy mildew looks like. So you can see our chlorotic tops of the leaves, right? And it kind of looks like a nutrient deficiency. But when you flip the leaf over, you have this sooty black spores at the bottom. So we set out to develop downy mildew resistant sweet basils. So how did we do that? We started the screening, and so essentially we started acquiring lots of different basil lines from all around the world: Asia, South America, the USDA, germplasm bank, Africa, commercial seed sources, and we started evaluating them in our fields. So here's an example of a susceptible sweet basil type and an non-sweet basil resistant type. And so essentially this is what our field will look like. We would go through our field and score these plants based on our rating scale here. And you would, every basil, sweet basil that we would have either would look like the Genova up here, highly chlorotic, necrotic lesions and severe sporulation, or like Eleonora, which is another sweet basil variety. And it's still very chlorotic, not commercially viable, but had some tolerance to downy mildew. None of the sweet basils looked like these these three on the left. And also, in order to screen faster and have multiple generations, we developed a high throughput screening method for the greenhouse. That consists of a dew chamber with 100% humidity and a misting chamber that would mist every 2 minutes. And essentially, you would collect inoculum, spray that inoculum onto the plants. You would keep it in the dew chamber for 8 to 12 hours. The spores would germinate, and then you would take the plants out, and over the next week, the spores would, or the Oomycete pathogen would colonize the tissue, and then once you put them back in the dew chamber, you would get sporulation like this. So after screening many thousands of plants, we identified this variety of that was resistant called Mirahani. Among others, there were many different species of Ocimum that actually had resistance. But through genetic diversity analysis, we identified Mirahani as being Ocimum basilicum. So we thought this was promising. And so you could see it doesn't look like Ocimum basilicum, and it was high in methyl cavicol, which is a licorice aroma. And we crossed it with our SB22 susceptible variety. So we had an F1 plant that was resistant, so it looks like it's a dominant resistance. And in order to identify how many genes control this resistance, we selfed it, we back-crossed to both parents, and we got all of this diversity. And we identified 2 genes, BDM1 and BDM2, that work epistatically to provide resistance. And so we set forth with this knowledge, and we designed a pedigree breeding selection scheme to remove these recessive carrier alleles that cause this susceptibility. Also interesting, we noted methyl cavicol, which is that licorice aroma that's very high in Mirahani, was actually dominant over the linalool, which causes the sweet basil aroma in SB22. When you look at the F1, you could see that the methyl cavicol was higher than the linalool. So after many breeding generations, we ended up with sweet basil varieties on the left, and this one's called Rutgers Obsession DMR, that were resistant, and this is compared to a commercial sweet basil control on the right. And here's another picture of that. So here's D. Genova commercial control on the left, and a Rutgers Devotion DMR on the right. And here's just some more pictures of this. This is a field trial, and you could see these 2 are commercial controls. And by the end of the season, they were just sticks. And then on the right here, you would have the Rutgers DMR lines, the resistant lines. And by the end of the season, full leaves, full canopy, and were still marketable sweet basils. Here are some close-up shots. This is Rutgers Passion DMR, and this is Rutgers Devotion DMR. And they were actually featured in Organic Seed Growers catalog this past year. And here's my advisor, Dr. Jim Simon. And this was a commercial organic farm. So Rutgers Devotion was actually successfully grown in an organic system in southern Florida, which in the United States has the highest inoculum density of basal downy mildew. So we identified this resistance and we bred for it, but we also wanted to identify quantitative trait loci that are associated with this resistance. So we created a mapping population with the Mirhani and SB22 cross, and we actually identified 3 sources or 3 QTL associated with this resistance that make up between 4 and 28% of the phenotypic variance. However, the resolution for this map wasn't high enough, so we actually could not identify molecular markers that were associated. So I've been working on updating this linkage map, and so I've actually developed a linkage map that has higher density, more markers per length of the linkage groups. We've also been sequencing the basal genome. So we took our SB22 cultivar and sent it out for sequencing, and we got 26 pseudochromosomes, which comprise 75% coverage of the total genome. And actually that makes up about 1.94 gigabase pairs of the basal genome, which the basal sweet basil genome actually has about 2.3 gigabase pairs. Also, interestingly enough, when we did a BUSCO analysis, which is just looking at the number of gene copies in the genome, we found that 88.2% of all BUSCOs were duplicated or were found in 2 copies, which is further support for the fact that Ocimum basilicum is a tetraploid and has 2 copies of all the genes. Our collaborators in Israel, the Cohen group, they actually worked on overcoming that sterility that I mentioned earlier in my talk about when you cross Ocimum basilicum and other species of Ocimum. So they did something called embryo rescue, where you pollinate the flower and you take the ovule that's fertilized, and then you culture it in tissue culture media. And then that tissue culture media essentially helps that plant develop fully. And they actually developed resistant sweet basil varieties using this technique. We've also been working on developing downy mildew lemon basils and downy mildew other like Thai basils. I'm going to quickly go over this. So we actually identified Ocimum canum species which came out of Zanzibar as fully downy mildew resistant. And through some lemon basil trials, we identified in some of these families 4% resistance. These are some of our breeding lines. You can see out of the 1,000 basil plants that we grew, only a small percentage of them actually had resistance. Something else that we've been focusing on is Perinospora bulbarii is an oomycete and downy mildew causing pathogen, and other downy mildew causing pathogens have developed new races, such as the ones on lettuce or spinach. We're already seeing some anecdotal evidence that mutations have been occurring because resistance is breaking down in our downy mildew resistant lines and the lines developed by our collaborators in Israel. They've actually identified a strain of downy mildew from Malawi that overcame their resistance. So in order to address this, where we plan to use differential panels where we have basils that have different sources of resistance, and you can screen those screen for new races with those panels. We plan to collect downy mildew samples and do a genetic diversity analysis. We also plan to identify new sources of resistance like the Ocimum canum that I mentioned before. And we could also use CRISPR gene editing technology to help further our goal of developing resistant basils. And we're seeking further collaborations with Basil, looking for new races of downy mildew. If you notice resistance breakdown, we're also looking for new resistant basils as well. And so This is the acknowledgement, so we can acknowledge the USDA and our other funding sources like the New Jersey Agricultural Experiment Station, our collaborators from around the world. This is my own personal acknowledgement section with there's always an army of undergraduates and graduate students who have been helping me throughout the way. And if you have any questions, you can address them to my email down here or my advisor's email, or you can contact us on social media. Our official Rutgers Basil account you can find on Instagram, Twitter, and Facebook.